So hello everyone. Thank you to offer Nanopore for the opportunity to present or part of our a program and application that we have at AdventHealth Orlando. I want to make emphasis that the application that we were able to develop was from the research use only for the MinION, the Flongle, and also for the GridION for our internal application as a clinical laboratory and a CAP accredited laboratory in the United States. This is the clinical use of whole genome sequencing as a cost-effective in-house approach for bacteria and fungi identification. My name is Dr. Jose Alexander. I'm the Technical Director of Microbiology for AdventHealth. I'm a medical microbiologist. First, AdventHealth in Central Florida is a large division. We have a main campus with around 1,368 beds, with an additional 7 hospitals with a total of 1,549 beds. It's a large clinical microbiology laboratory with 75 medical laboratory scientists, 2 managers, 7 supervisors, and we handle an approximate of 2.5 million samples annually. We perform a lot of traditional microbiology testing. Bacterial culture, we have access to instrumentation as the MALDI-TOF, the VITEC system. We also have access to some manual susceptibility testing for diffusion or bromide dilution. Mycobacterial culture, AFB, that we do a lot of samples annually. We used to set the plates, grow the organism, and send them out for identification. And fungal culture, where we also perform a large amount of them every year. We perform morphological identification. So for years, we use the for mycobacterial identification, we use this instrument called the GenProbe, able to identify a group of organisms, a group of mycobacteria, specifically TB and MAG were the most predominant, and the one that was the high prevalence, they are the high prevalence for us. But this instrument was discontinued in summer of 2022. So at this point, the identification of TB and MAC, that was our main use for this platform, have to be sent out to a reference laboratory. So beside the non-TB mycobacteria, now we have to add the volume of this group of organisms. Now, we were able to develop an application for whole genome sequencing since we were evaluating multiplex PCR or whole genome sequencing that allows also to expand beyond mycobacteria through bacteria and also to molds. We have a send out volume in 12 months in 2022 for bacteria ID. We send it to Mayo Clinic. We sent around 93 isolates in a year. These were isolates that were not identified by MALDI-TOF or VITEC with an annual cost of $33,800. Mycobacterium ID, we have an annual volume of 667 isolates sent to National Jewish with an annual cost of $249,000 with an average turnaround time of 9 days. So in total, for these 12 months in 2022, we send out 760 isolates with an average cost of ID plus shipping of $368 per isolate, with a total annual send out of close to $280,000. So that was a significant operational cost for our laboratory. We start evaluating different approach, and the Oxford Nanopore was one of the main one. The reason of that is, well, the simplicity and of the platform is able to the platform will be able to be implemented in a facility that don't require a high complexity for a molecular diagnosis. So there was a small device, the MK1C. We start with 2 of these devices, and with a manual process for sample preparation, extraction, and library preparation. This process has been currently automated, and we now have access to a GridION, and we use heavily Flongle flow cells, and MinIONs has been used lately due to the increase of volume. So when we implement the NGS, we evaluate a framework and a requirement. They have to be implemented by the microbiology laboratory instead of the molecular lab. Have to use microbiology standards. For example, to prepare the dilutions, we use the McFarland, just like we do for susceptibility testing. Have to be reliable, capable of multiplexing. We are able to multiplex from 2, 3 isolates all the way to 24 or even higher. Rapid turnaround time, have to be 24 hours from the isolate to the final ID. They have to be cost effective. How you saw in the previous slides, the cost of send out was around $360. So have to be less than the $350 per isolate to be cost effective for us. Have to be simple. The hands-on time have to be less than 1 hour. Minimum support instrumentation. So we need to have instrument that could be flexible for multiple application and something that doesn't require a large footprint. Have to be comprehensive, simple, and intuitive pipeline. So we don't have access to bioinformatician or genomics. So we needed to be able to have access to third-party pipeline, something that could be easy used by our scientists. Traditional application in NGS, especially for diagnostic, is typically in large academic centers. So the microbiology laboratory receive all the traditional microsamples. Some of the requests for NGS that are done in-house are sent to a research or molecular laboratory with molecular expertise, and then the result is being able to be sent that to the microlab for reporting. And the approach that we have at AdventHealth was we need to be able to have this expertise and capability inside the clinical microbiology lab. We needed to make sure that this test is done by clinical laboratory scientists. They understand microbiology. We take NGS as an application, as a diagnostic tool, not as a specialty. Just like we have expertise in the MALDI-TOF, in susceptibility testing, in multiple immunological techniques, NGS was one more of those techniques. So we start with an activation, especially for mycobacteria, a similar approach for molds. We get a concentrate from the midget, that the BD broth for mycobacteria, or 2 colony loops from solid media. We perform inactivation using ethanol and sonication. And before we start the validation, we need to make sure that these organisms are completely inactivated. So we use a pre-validation inactivation process before the validation. We don't do this routinely anymore because we were able to prove using multiple species of mycobacteria that this process were able to inactivate the organism. This is done under the biosafety level 2+ or 3 from the mycobacterial laboratory. Then the final tube with the organism inactivated can be removed from this room and be worked on the molecular side. This tube is placed in a cell lysing where we produce with a bead beater. We release the DNA. The DNA is extracted using the isopure mini and the Zymo kit. Library preparation, we use multiple library preparation. Could be the rapid or the native barcoding. We use the flow cells depending on the amount of organism. It can be the Flongle or the MinION, sequencing in the GridION, and we use the epitome as the main database. When we don't have organisms that identify in the database, we use the one codex. The whole hands-on time is less than 40 minutes. We sequence for 22 minutes, and the data analysis probably take 20 minutes for all the isolates. So in total, we have an approximate of 22 to 24 hours from beginning to end. So this is similar process for all isolates, bacteria, mycobacteria, and fungi. The only thing that is different is the sample preparation at the beginning of the process that can vary based on the organism. The sequencing parameters, so we sequence 6 actual isolates in a Flongle. We want 2 QC, sequence for 21 hours. Positive and negative QC have to be positive for an acceptable run. More than 5 reads, except for the negative QC, have to be detected in every isolate. And the threshold per isolate has to be a minimum of 5 reads, a relative abundance greater than 3%. And if it's more than one organism at 3% or more, we have to evaluate the possibility that we're dealing with a complex or a group. And the total cost per isolate is currently at $63. The validation was done with 120 acid-fast organism, AFB, 105 AFB, 15 aerobic actinomycetes. 20 run with 6 isolate plus 2 QC. So the 20 QC were taken as negative, a true negative. We evaluate accuracy and precision using different species, reproducibility between different platforms. At the beginning, we used the Kraken 2 platform. That's something that we don't use anymore since the epitome now is based on the Kraken-2, and we have access to multiple other databases. The interference substances with multiple media were evaluated, and cross-reaction between abscessus group species from this particular group to make sure that the accuracy and reproducibility was appropriate. In the total, we ended with 117 organisms that were identified. And the reason of that number is because they were present on the 3 databases. 3 isolates were removed because we're not in one of the databases. 16 out of 16 abscessus were correctly identified to species level, and we have a good reproducibility of 9 of 10 organisms. One of them was not present in 2 of the databases, so that's why we only have reproduced 9 of the organisms. We have 100% of technical accuracy. That means if the organism is in the database, we were able to identify. So in total, 98% of all acid fast and partial acid fast that we currently isolate across AdventHealth in an annual base are present on the database. So this is important because if the organism is not in the database, we don't consider the process that failed. What we consider is that the database needs to be updated before to be able to get to that conclusion. So the technical accuracy is critical because we allow us to understand that we are able to identify organisms to subspecies level. When we evaluate the post-implementation, especially in 2023, the bacteria ID, we have 78 isolates. At the beginning, the first time we have around 90. Again, this is those that fails MALDI or VITE. The annual cost of only bacteria ID in-house NGS was around $4,900. Mycobacterial ID, we have 705 with an annual cost in-house NGS of $44,415 and a turnaround time of 24 hours because we do it overnight. So you can see that the total in-house NGS now, we're 783 isolates with a cost per isolate of $63, give us a total of $49,330 annually. This is only operational cost. This cost can get close to $100,000 if we consider the implementation with instrumentation for the first year, but only operational was $49,330. So what was the impact? Well, a reduction of turnaround time, 9 days versus 24 hours for identification. That's a significant impact on turnaround time, especially for mycobacteria and rapid approach for empirical treatment. Full control of the process. So now we are able to isolate and decide based on our data which organism need to have identification. It hasn't been done before. And also based on sources. Reported subspecies and complex group is possible to the abscessus group and other species, other complexes. So that allows us to be able to differentiate abscessus from meseliensis that we know that's significant for treatment impact of the organism ID. And the cost saving. For the first year operational saving was $230,000, so close to $250,000 saving only for doing NGS in-house. So the question is, how can we justify the use of NGS, right? See, sometimes we don't have in the US a CPT code to be able to charge or to bill for these tests. Well, as an operational cost, if your laboratory use reference laboratories, this is an important step to reduce the impact of the cost as an operation of your laboratory. So I think this is a significant improvement of operation and also give us the capacity, the capability for having this technology in-house. This was our pre-NGS method identification. For organism, for bacteria, the first method was MALDI. For mycobacteria was a send out because we didn't have any other method. And for fungi was microscopy. And second method for bacteria and fungi were send out because we didn't have any other method. Currently, we keep the same process for the first method, except for mycobacteria now is whole genome sequencing. And for bacteria and fungi, whole genome sequencing is the secondary method if a MALDI test fails for bacteria or microscopy is unable to identify the fungi. We have multiple other applications. Besides the bacteria, mycobacteria, fungi ID, we have a positive blood culture ID is identifying the organism directly for the positive blood culture when the organism doesn't grow in solid media or the BCID fail. We have a strep pneumo serotyping genotyping application by whole genome sequencing. We do epidemiological surveillance when we have a potential cluster to differentiate this organism before doing a full epidemiologic investigation. We do AMR analysis for unusual or complex resistant mechanisms. We are evaluating a plasma cell-free DNA application, a sterile fluid, paraffin block, and a fungal rapid NGS directly from sample to replace a fungal culture. So in summary, NGS for infection disease diagnostic must be considered a tool and not a specialty. Clinical microbiology expertise is important. Implementation and solutions should be optimized for clinical microlab. It is different oncology laboratory and molecular research lab from a microbiology lab. The flow of samples, the complexity, and the time that is required for this result to be back are critical. The research use only and guidelines are needed for adoption beyond academic and research facility. Being able to prepackage an NGS solution with RUO, with process not only for the sequencing, but before the sequencing and after, and being able to offer third-party databases, easy for laboratories to use, are critical, because in that way, laboratories can use a lab-developed test, can validate a lab-developed test, since many laboratories are CLIA or CAP accredited. And importantly is, please do not discontinue the Flongle. Low volume and cost-effective flow cells are needed and important in microbiology. Sometimes we have 30, 40 samples and immunogen is excellent. But sometimes we only have 4, 3, or even 2 samples, where the best and the cost-effective solution for us is using a small flow cell as the Flongle. So this is an important tool for clinical microbiology laboratories. So replacement of the tool of the Flongle, but having access to that type of flow cells is important. This is our AdventHealth NGS clinical team. So we have a morning shift that do library preparation, sequencer loading, and data analysis. We have a group of 5 clinical laboratory scientists. And the night shift, we have a team that do the sample prep and amplification for targeted and metagenomic NGS. You can see that our NGS is just a small portion of a molecular room. It's a small corner. In this picture, we had the MK1C, but now we have replaced that for the GridION. And everything happened in this little corner with support of some instrumentation that we have for clinical diagnostic that can be also used for additional steps during the NGS. Thank you very much for listening and for the opportunity for me to present it. And if you have any question, I will be happy to answer.